This is a production of Cornell University.
Yes, no, I'll be brief, but yeah, it's my pleasure to introduce the topic today, being presented by Star, Sage, Greg, and Mathew. They've been, working with the Innovation Lab for Crop Improvement. I've been really kind of engaged in interacting with them since the start of the program. One of the stated goals of the Innovation Lab for Crop Improvement is to develop tools, technologies, and methodologies for crop improvement. And in my experience, developing these tools, technologies, and methodologies is relatively straightforward, but developing, these TTMs that are actually adopted and have real meaningful impact in a breeding program is incredibly difficult. And being able to really track the impact that these tools are having can be really, a challenging endeavor, especially when dealing with, large and complex research, organizations like a lot of the NARS that we're partnering with ILSI. And it's been great working with this team because they're bringing a lot of expertise that we don't really have, certainly in the plant breedings and genetics section. They've been able to put together some great surveys that help us get a better understanding on, really how these programs operate, what some of these baselines are. And this is really just incredibly important information as we try and actually develop collaborations that are going to be impactful with these organizations and try and track and learn from, how these tools are being used in the programs. And so with that, I just like to thank everyone for presenting today, and I'm really looking forward to this presentation. Thank you.
Okay, thank you so much, Kelly. Yeah, all right. So a quick outline. Sage, Greg, and myself is going to have three parts of today's presentation. We'll talk a little bit of context objective of these surveys, and then we'll give you two overviews, the methodologies and some of the findings from the surveys. Actually, originally I was going to put 3 surveys, and I don't think there's enough time. And tomorrow I'm going to present that in the live meeting with Kelly and Guar's lab, and I will cover my recent interviews with all the area objective leads. Okay, so today only the 2 surveys, and at the end we'll share some of the learnings, and then also mainly is for discussion, very informal, and then entertain your questions. I think for ourselves, we are planning the next phase of the grant proposal, so allow to learn from the discussion as well. Okay, so the quick begin. So CGIAR and the NARS is the main topic today we are going to speak. You may wonder, as Cornell community, why we care about the CGIARs, why we care about NARS. In the developing world, this is the largest public breeding programs, agriculture, and funded by many funders, and it's so important because since the Green Revolution in the 1960s, the solving the hunger, and even in today, in terms of managing the climate change. So this has a huge impact. It's not only from the innovation perspective, it's also in terms of adoption on the farm's field. And this is a great model, has a large footprints over hundreds countries. And Cornell and the USDA, we are one of the 3,000 partners. So for the graduate students, you are here, your research, your publication, even your future as your different opportunity to work in industry or academic or funding agencies, one way or the other, because you're in plant breeding, in plant science, you'll be in this value chain somewhere. So that's why so many people we know on campus work with CGIAR and NARS. So you can see here 15 centers, they are formed over since the Green Revolution over time, and sometimes there were 17, 18, and sometimes 13. So right now, active 13 centers. They lead agriculture, innovation, technology transfer to small holders in the developing world. So they mainly serve these, is not really for the very profitable, mainly yield, like large multinationals. Huge inputs that also cause today's climate change challenges. So the small stakeholders meeting the local nutrition and also diet and equality, this huge impact as a model in addition to what we learn from them. So this is one recent from the last 2 to 3 years, the genetic innovation from CGIAR, the pipeline from the left to the right. First part is product design to figure out what farmers, what these customers, what type of products they need. And this involves economists, market intelligence together, looking at from the end, begin with the end for local area adaptation to help design and going to either the center part as the gene bank of pre-breeding and then go to the breeding networks. Breeding networks, they may cause from original breeding crosses go with population development and early testing, late testing, all the way until on-farm trial testing. Okay, so this itself can take, depends on the crops, can last somewhere from 4 to 8 years before they hand off to the seed systems in the product delivery, in the variety registration, etc. So this is a little background from the CGIAR perspective. They work in the broader network with the NARS. So I'm going to pass this on to Sage, and I will come back when we talk about the second part of the survey. So with that, Sage, please unmute yourself. Thanks.
Great, thank you, Star. So as mentioned, I'm Sage. I'm a research support specialist at the Tata-Cornell Institute for Agriculture and Nutrition. I'm joined today by my colleague, Greg Traxler, who is a Tata-Cornell Fellow, also working on the Feed the Future Innovation Lab for Crop Improvement, or what we call ILSI. So we're both members of the ILSI Institutional Capacity Research Team. And so just to know, we're mostly a group of economists. So we're really excited to present some of our preliminary results and ideas with this group of plant breeding researchers. And Greg will be available for questions as when we get to the Q&A session. So I'm going to start by just giving a background of ILSI, which is hosted here at Cornell. ILSI aims to advance crop improvement programs in developing countries by facilitating global communication through a network of experts in national agricultural research systems, or NARS, and U.S. universities, prioritizing gender and youth inclusivity, nutrition, market relevance, and crop resilience. ILSI is designed to support NARS to set breeding priorities and adopt modern technologies in plant breeding research. Regional centers of innovation in East, West, and South Africa, Latin America, and the Caribbean enable the NARS to collaborate and share resources. The project focuses on orphan crops that are relevant to local diets and adapted to environmental conditions. So the Tata-Cornell Institute is tasked with measuring the change in institutional capacity of partner NARS as a result of the ILSI project. Next slide, please. So here I'm showing the simple theory of change. U.S. university scientists comprise the upstream research areas in genomics, phenomics, breeding informatics, and priority setting. And then with the objective areas of research, there are tools, technologies, and methodologies, what we call TTMs, that are passed on to NARS through various trainings and workshops with the expressed outcomes of increasing the rate of genetic gain to impact economic growth, resilience, and nutritional development goals. Next slide, please. So this is a little bit of a more in-depth framework for the ILSI project. The ILSI priority setting objective areas working with NARS to implement various studies to better understand and incorporate stakeholder input to create product profiles. The trait discovery team is tasked with translating those product profiles into feasible breeding objectives, employing hypothesis testing and experimental design. The genomics, phenomics, and breeding informatics team have a more direct impact on the breeder's equation components, enabling breeding programs to employ technologies to reduce cycle time and streamline the breeding process, ultimately leading to genetic gain. So ILSI's philosophy suggests that if target population needs are optimally understood through effective priority setting and integrating gender equality, youth engagement, nutrition, and resilience, varieties will be adopted effectively, thereby meeting their intended impact on livelihoods. Next slide, please. So by surveying the NARS partners, we want to better understand the crop improvement system. We also wanted to know which modern TTMs are being used in which breeding programs. And by identifying capacity gaps, we could better understand the strengths and needs of institutions and individual breeding programs. And then lastly, monitoring capacity changes over time enable the team to understand which ILSI interventions have an impact on capacity. Next slide, please. So to come up with the metrics in a survey tool to measure institutional capacity, the team first conducted a comprehensive scoping review of the literature to define institutional capacity in the context of plant breeding. We identified 74 papers that assess the various components that make up institutional capacity, and this helped us create a list of indicators. We then reviewed existing capacity assessment tools like the BPAT, which was used in the CGIAR system, to identify and adapt relevant metrics to the ILSI context. This led to various discussions with experts in the field, including ILSI objective area leaders, enabling the team to arrive at this initial list of survey questions. We conducted a Delphi among experts and stakeholders to generate a consensus on our list of questions. And then using this list of questions separated into indicator areas, the team conducted a pilot survey with 14 ICRASAT breeding programs in Africa and Asia to test the functionality and technical features of the survey, and also to receive some feedback from the programs concerning the quality of the instrument. We then went back with all the feedback and simplified the tool and reduced questions to eliminate redundancies and streamline the survey. Finally, we conducted the survey to measure the baseline capacity of 18 NARS breeding programs involved in the ILSI project. Next slide. So on the left shows the components of capacity that were identified in our literature review, including the various resources available to a program, management and planning, collaborative activities, breeding activities, setting priorities, and stakeholder involvement. These components were translated into our indicator areas for the survey, as seen on the right. And in each indicator area represents a module in the survey, each containing about 10 to 15 questions. Next slide, please. So we surveyed the 9 ILSI NARS partners with a total of 18 breeding programs in Africa and Latin America. The programs surveyed were working with crops including cowpea, sorghum, millets, common bean, and sweet potato. Next slide, please. So on the left, you can see that about 30% of breeding programs surveyed did not make any crosses in 2019, and only 6% made more than 100 crosses. So most programs made in between 1 and 100 crosses in 2019. And then the graph on the right shows variety release over a 3-year period. Of the programs that reported to release varieties, we can see almost 50% did not release any varieties in 2017 to 2019. So only 34% released 3 or more varieties over that 3-year period, corresponding to an average of 1 variety per year. Next slide, please. So as for the use of modern technology, we first looked to the digitization of data, where we found just over half of the programs surveyed did not collect any phenotypic data digitally at the field level, while 27% of the programs collected 50 to 100% of phenotypic data digitally. The right table shows the percentage of programs that use breeding management software for various breeding activities. So using software for trial designs or trait data were the most common, while very few programs use breeding management software for genomic data. On the bottom left graph shows some of the more modern techniques to achieve greater genetic gains. And we can see that half of the programs use genomic services, a third use genomic assisted selection, a third use marker assisted selection, and less than a quarter use genomic selection. So since achieving genetic gains is the express goal of the ILSI project, we then asked NARS if they measure genetic gain and learned that just 11% of the programs that were surveyed do measure genetic gain. Next slide, please. So as I described earlier, resource availability comprises an important part of institutional capacity, like the availability of scientific staff and genetic resources. And we learned about 60% of the NARS surveyed have less than 3 full-time equivalent staff available to their programs, and most of the programs have less than 500 accessions available to them. For comparison, I'm displaying the data from our pilot survey with ICRASAT breeding programs, where most of them reported to have greater than 1,500 accessions available. Next slide, please. So some things that we learned is that there's high heterogeneity within the NARS, and this should be considered when targeting TTMs that will be most appropriate based on the NARS capacity. So most NARS were making less than 100 crosses, released less than an average of 1 variety per year, and had a small number of scientific staff and genetic resources available to them. At this level of plant breeding research, there's a smaller probability to enhance rates of genetic gain. So in contrast, there are a few programs that were surveyed that are reporting to use modern techniques, conduct many crosses, and have more available resources. Next slide, please. A large component of institutional capacity is the policies in place that determine the funding allocated to each program, which impacts the availability of resources. So orphan crops comprise a smaller proportion of institutions' budgets compared to other crops. Given that there's these institutional constraints in the state of breeding programs, within the state of breeding in many of these programs that we surveyed, that I was describing earlier, it may be best to adapt capacity building efforts to ensure that NARS breeding programs are testing and releasing varieties more effectively. So that could mean targeting TTMs such as collecting and analyzing stakeholder input to generate product profiles, more accurate phenotyping, data management, and experimental design. Then the programs that have a higher level of capacity will be better candidates for targeted training of these more advanced genomic type TTMs. Next slide, please. And then lastly, tools like our survey to monitor capacity can be helpful in the decision-making process to better understand each program and adapt TTMs appropriately. So just a little background that we will actually do the same survey at the end of the project in this upcoming year to measure that change in capacity. So we can track those changes over time using this survey. And very few NARS are measuring genetic gain, variety turnover, adoption, or yield gains in farmers' fields. And these are really important measures of progress and to track the actual impact on plant breeding efforts. So this is a consideration of what we might want to quantify in the future. So I'm going to now hand this over to Star again to talk about some of the studies that she's been conducting. Thank you.
Okay, thank you so much, Sage. I know she ran through very fast. Many of you may have questions. If there's just one quick burning question, we can also save some time at the end. Anyone? Okay. If not, I'm going to switch to the second gear. All right. So this is on the CGIAR. The other ones on these two completely independent survey. So this survey was conducted in the first 3 quarters of 2022. The context of this survey is with the 1CG, means the 15 centers, there are similar crop breeding programs across different centers. And the 1CG was for them to work together as these large companies located in different parts of the world. With these different centers have very limited bioinformatics resources, including the institutional capacity, servers, data management systems, also their statisticians, quantitative genetics, etc. In this context, the thinking is if all the bioinformaticians, quantitative genetics, they work together with the COVID mode, people can work online and can provide services support to the centers, the regions. They do not have the resources in the data analysis, in the decision support, etc. And then they survey with this in the mind, there was also transitional in terms of people from the excellence breeding to the breeding resources and genetic innovation, these new funding as a project, the new program. So Yunhua Li, she took the position. Kelly was before as the module 5 lead in excellence breeding, and she led the biometrics and informatics. So I worked with her as the program manager, managing all the bioinformaticians, quantitative genetics across all the centers. And while this funding transition, the Module 3 lead, the genotyping, the Service 2 manager, Yingwa, and also Eduardo, who's the Module 2 lead from Excellence Breeding. So together, Yongwa, Module 5, the four of us in this transitional stage, we're trying to understand from all the breeding program perspective, are they ready if we have a centralized service? And also, what are the key common demands if they are ready? So that's the background. So as I mentioned, there are two overarching goals. The first one on the left-hand side is, are they ready? And then from technical perspective, also psychological willingness to how everyone work together. The second one is the common demands. If they are ready, what type of capabilities they need for these quantitative genetics bioinformatician to provide. So the survey had a total 8 pieces, and I'm going to get into more details later. So in the next several slides, I'm going to talk about the methodologies we used, also some of the results, and some discussion about the insights. So these all together in these two overall arching goals. So the first part is for social scientists, I think many of you already know sample size to collect these type of priority setting. And then for us, Stanfield, we were learning who do we send a survey to? The data is going to really indicate truly the program what they need rather than diluted. So with a lot of back and forth, the four of us thinking, and we agreed with the different CG leads is we only send it to the head of the breeding program so they can work themselves and distribute if needed. And this need to be really representing how the weight from what they need instead of we send to everyone. That's why highly selected. One survey sent to each person with personal email. We wrote describing the background, why this is important, and also time to turn around, etc. Individual survey sent to NAR breeding, NAR CGIAR breeding head. And it turned out some of them answered themselves, and they also allocated to their subject matter expert for the parts of the questions they are the best expert to answer. We sent to the 12 NARS breeding head, 6 responded. And then among total 25 respondents, they represent 16 different crops and 12 centers. And this is the overview for the all the CGIAR people who answered. So you can see the center affiliation and the crops they represent. And the ones highlighted in red, they come in answering this survey with the manager role, looking at this big picture. And the other one unhighlighted, they are almost like lead breeders representing their entire program. And we have 89% responses rate and 200 almost 40 participant rate because some of the centers, they have multiple subject matter expert answered the questions. So from NARS, interesting, we sent it to 12 breeding head, but only 6 responded. So we can discuss later why we couldn't accomplish the same participation. So here's a 50% participation. And then, so for respect, I'm not listing the other 6 we invited, but didn't answer the survey. And hopefully this also represent the 50% of the NARS breeding programs need as well. So hide this. So the first question, as I mentioned, overarching goal on the left-hand side, we want to know is, are they technically ready to measure the institutional capacity? We want to define what's called institutional capacity, right? So capacity including technical and human capital, resources, germplasm, etc. Together, collectively, are they able to do the program quickly and in good quality to meeting the demands and the mandates So this is how we define as the institution capacity. So first we want to understand, are they ready if we want to provide these centralized services? So from here, I don't know, you can see very well, but overall, we want to ask them very similar, these two independent studies as a SAGE, and we are thinking, good heads think about the same thing. We ask them for phenotypic data or pedigree data, genotype data, and breeding crosses, these different type of data, are they already captured in digital format? If they are digital format, they are shared in centralized servers, then these biometricians can sit somewhere, analyze their data together and distribute the results, and that will give more power, right? So we want to ask them, are they digitalized or they are just doing on their own computer or on piece of paper, etc. So 75% of the breeding head or the participants, they mentioned they are ready. They already have these in digital formats. And the phenotype data mostly digitized, followed by genotyping data and the pedigree data. And lastly is the process. This means SOPs, how things make transition from one person's hand, one stage gate to another. So that's at least digitized. And here I want to point out is with the data, how you really get insight is not only you collect data, because this is harder to see what does that mean, right? So we ranked. And we also developed a method to look at it is called sum of product as the indicator to look at this with more insight. Basically, the degree of data digitized measured as a percent of data collected and also stored in the digital formats. And you can see the phenotype data when they ranked as low, like say 0 to 33%, mid 34 to 66%, high means 67 to 100% data already in the digital format. And when you multiply the data level for each one ranked by the counts, and you sum that up called sum of product, this is as one indicator. And with this, you can more easily, simply compare them. So you can see the breeding process only 40%, 46% digitized with the highest at phenotypic data. So that also match with what we see in SAGE survey as well. Another thing we asked them about the infrastructure. Do they have the infrastructure, cloud or server? And are they ready, even they have the infrastructure to share, they are ready to use the data analytic services. So when we ask these questions, just very simple for them to read as 1 to 5. So the current state readiness will qualitatively assessed at a low, medium, high, or as 1 to 3 or 1 to 5. From 1 is lowest, 5 is higher. This is qualitative. It just gives some indication. It's not directly quantify their readiness. But overall, you can see the ones on the right side, infrastructure, actually, they are pretty good for most of them. And the 5 is the highest here. And in terms of readiness, actually, more the 4 and the 5, they are ready. They want to have the centralized analytical support. One is the server, another one is the data management, also the management practices. Are they willing, right? So we asked them about currently, do you have your data just dispersed, means on your local, or they are like a hybrid? You have some centralized on cloud server or purely centralized server. So what we see is in the 3 means the centralized 24%, and large majority 64% still on their local computer. These definitely making sense. You can see very diverse differences. And the 24% program, they are technically more ready to adopt these centralized services. So transition into the next part is in the goal 2. The second goal is about if they are ready for centralized analytic function to support them, then what kind of capabilities do they need from the phenotypic, genotypic, bioinformatics, and data management, our full functional area. We asked them the features they want to have as a must-have, or should have, could have, or will not have. So the must-have means non-negotiable, and should have is important, and if but not adding significant value. And could have means nice to have, but if we leave out, have small impact. will not have means not at this time frame is important. So we ask them to put these capabilities in these buckets, and we also assign a numerical value 4, 3, 2, 1 for each of the category. This allow us to quantify the final demand and also find out the priority ranking in the capabilities. So you can see this is just one example. In terms of data collection, after collecting, you do data curation and data analysis, decision support. In these 4 functional areas, what do they consider as must-have? And based on the counts, the frequency distribution, you can see this is a way for us to quantify it. So the demand calculated not only with the must, should, could, and will not have. We also did a sum of product, very similar like early on I showed you in the previous slide. The ratings for each category, the ranking multiplied by the number of accounts, and you do the sum. This gives you a cumulative number, easy for you to compare them. And then to simplify it, here you can see the decision support is the last stage of the breeding analytic to provide for product advancement. Consider is in the highest demand versus data analysis, and the lowest the data collection. Basically, they are saying they can take care of the data collection on their own, but they need a support for decision support. And then you can also see for the must have, should have, and will not have, some centers have a little bit diverse views. For example, for data collection, for one center, they think this is very important, etc., and data curation. So for phenotypic analysis, what kind of capabilities they need, they agreed on based on the analysis here is optimize the trials, the design, and also the product advancement. Basically, there's not much difference. You can see the points, and the relatively lower at the end is like for some program, for example, they think should have is a site selection here. And then for some program, they want to have the variety catalog, germplasm catalog, to compare germplasm. And then for 2 of them considered very important, but vetoed by other people participating in this. That's why at the end is as lower ranking. That's the phenotypic. And how about for marker system selection using mid or low density genotyping based analytics? We see, I think Gary Atlan, if he see this recording, he will be happy. He always talk about calculate the GRM, the GRM, this rise up, showing this the highest in demand, and followed by the pedigree verification. So for mid-high density genotyping, here is the genomic selection or GWAS come in place. We can see GWAS considered not important for the breeding program. Instead, predicting the breeding crosses to go to their crossing block considered the highest importance, and followed by predicting GBVs. And the third function is the bioinformatics in terms of capabilities, infrastructure. We can see provided infrastructure. That's what very timely they need. And then followed by merging data from different sources, including phenotype, pedigree, genotype, etc., and across the different actors, merging them together. That's what currently repeatedly saying in the 7th, 8th section of the survey. The 8th part of the section, the survey is about how the data flow integrate together so that allow them working holistically from the pipeline A to Z. And here they talk about the digitized processes. This come in is very obvious, and also centralized databases so that they can share with programs in different regions. Okay, so this is the main results. And here the summary I already mentioned, based on their ranking, the common high demands for the second part of the goal, we understand. And with these high demands, now we have to ask about, do we have the personnel, the quantitative geneticists or the bioinformaticians to provide all these capabilities in high demands? are we able to give them the product or services? And that's the story actually continue to be discussed, but at least now we understand the breeding programs, what they need. And then key learnings, there are many, many learnings for myself through doing this with the 1CG breeding informatics team. Technical readiness considered really important. They have digitized data processes, centralized data storage for sharing. And then from here, from SAGE report, you can see as well, increase the investment. This uniformly from both NARS and CG Center, we need this to enable them to do the centralized data sharing. And the common demands, there are many, but in order to provide this, minimum viable products have to be focused on as well. That's where the phase currently, we originally wanted to build that, but that didn't take off. There are a lot of factors we can discuss offline. And but so many learnings how to make this ready for the early adopters. As you can see, some of the program, 24%, they have the data management ready. And for them, different stages of the tools they need, for example, making the breeding crosses at very early on. And very diverse programs. Sometimes, even though you have this high demand, when you go to talk to these breeding leads from different centers, they do not want to give away their own bioinformaticians. They all work together, support centralized, because different time of the year, different capabilities needed. So the resources and how to have tailored support and solving this bottleneck one at a time. So with that, I think Sage and also Greg, we can just open up for the questions, and especially in the context of planning the next steps with LC. And I think there's some good news. 1CG-NARS just got the new commitment for the next 2 to 3 years, almost like 900 millions. That's what I heard just 2 days ago. And hopefully the second part of the survey, we have to pause, and also from the people here on the line, and also here to see how we can be part of this value chain as a service provider or as the stakeholder. You see, you can contribute. Yeah. So I'm going to open up for the questions because we have a few minutes. And also just want to give a big shout out to the LC, the area, objective area lead. I did a focused group interviews recently. Not enough time to cover today, and tomorrow in the live meeting I'm going to cover. Also, thank you so much, Sage, Greg, and also Mathew for collaboratively. Recently, we are planning the ongoing survey. Yeah, okay. So I see people on the line. Okay. So yeah, Marjorie, please.
Do you have any ideas of how to increase the engagement from the NARS and their participation in the surveys? Or have you noticed any specific reason why the CGs are more responsive than the NARS?
Yeah, I think Sage and Greg and also add comments as well. From my part, I knew we sent exactly the same personalized email from our side of lead, one-to-one to the breeding lead from NARS as well. I think it's just nice direct working relationship. Maybe went to their spam folder. Maybe they didn't see the importance. So it's so important. We are very selective and also have the personal connection to raise the awareness. Yeah. Sage, Greg, from your surveys, you may also have the struggle about getting people's engagement participating, right? Especially the data. Yeah.
Yeah, sure. Yeah, that could be a bit of a struggle. Our advantage is that we're all working on a project together, that are receiving funding from the same source. So there was a little bit more of a buy-in from the NARS we were surveying because they were part of this project already. So, we all represent different teams. But we have tried to survey other programs, both in the CG and the NARS, and we've had difficulty getting people to respond at times. So yeah, these are things we're constantly thinking about, how to get people to buy into this idea that institutional capacity is a valuable thing to measure and track. And so that's, doing presentations and trying to talk to more people. And I think these personal connections make the big difference. We've had some responses based on, knowing some of the NARS from this program and then asking them to ask some of the breeding programs within their institution themselves. So yeah, this kind of collaborative network is really useful.
Yeah. Okay, there's one question from Kelly on the line. Kelly is asking, the surveys self-assessment versus these external evaluation like a BPAD, any sense of how well the self-assessment, external assessment align? So there must be some part align and some part may not so align because from some, yeah, go ahead, Greg.
Yeah, Star, thank you. We did include in our survey subjective questions of the scientists. How well do you think you're doing in particular areas? What's your subjective opinion on your capacity in data management or given areas? And then we also collected objective data. So we do have both kind of the counts of number of crosses and access to materials, et cetera, and then can compare those to the scientists' expressed opinions about their capacity. So I don't think that we've pulled those out to compare them, but we could do that.
And just to add on a little bit, something that we had done in our research effort is we visited some of the breeding programs and did some field work there. And with those programs, we conducted a midline assessment, and that was the major goal there, is to validate some of our findings, which had changed a little bit. We found that, one program that had claimed to be doing marker assisted selection, when we asked them in person, they weren't actually implementing that, or maybe they had markers available but they weren't using them for selection. So there can be some inaccuracies with this, but continuously monitoring over time and going in person has helped clarify some of those and help us to explain the questions and ensure we're getting more reliable data based on how we pose the questions.
Yeah, just want to add that actually the right questions to ask and also really design a question is more than half of the survey research itself, because what you get from the answers largely depend on how you ask this question. So definitely it's really a good complementation for on-site visit to the external evaluation, because even, for example, the answer they answered, like read from 1 to 5 level, right? If the breeding programs located in so many places, there is a huge heterogeneity. It's difficult to have just one number to rate it. Even the breeding lead think they are ready or they are already at the highest level. So we cannot really overlook the diversity, the how heterogeneous of these programs. So this is a quantitative, simple way to have a quick look. Yes.
I'm wondering how centralization of breeding analytics tools might affect the capacity building of young scientists at NARS or other breeding specialists working at the NARS. And I'm wondering if that came up in the survey at all, and do you think that would affect willingness to move to this model of centralized services? For example, if there's young scientists or students working with these NARS partners who are wanting to learn genomic prediction or wanting to learn database management.
Yeah, this definitely is going to be more difficult for the NARS than for the CGs. And then for the young scientists, I think the model is these on-site visit, hackathon, and even during COVID, this team work online together. The community building, we had one platform called a Breeding Informatics Analytics platform. So people online all join in different functional areas. And then they have different levels, some very advanced and some just beginning, and also they complement each other. Some knows Python, some knows R very well. So actually I see even for Cornell University and for other LC partners, we provide a huge value in this system as well. So for example, we have the NARS partners recently visiting from Costa Rica in ICE group for several weeks, and then also the one from Senegal as well. So they are learning, and then the university like us provide huge value. And then also the graduate students, right? Because the publications from the faculty here, and you guys are at the front end. So a lot of the learning here for you to dive into some of the case studies working with them. This could be a one model, because these young junior bioinformaticians, some of them finished the master degree, and then some of them actually pursuing. And after they find the opportunity, they leave from where they work, they come to US to do PhD study. We lose people over and over by working with them. And it's very difficult for the team there to hire, because where they are located, sometimes no electricity, no internet, and then they are asked to do this, to do that. It's not really easy. So more centralized resources for them to continue, even like seminar like this, allow them continue learning when they get stuck. The other is the infrastructure. So this platform is huge to how this is not only social capital to boost the morale for them to work, learning informally in this platform is huge. So may not be easy for the supervisor to say centralized, because they have a lot to do, but in the effectiveness, the collaboration, partnership offer great value. Yeah, sorry, I don't know this long answer. Okay.
Could I just jump in there for a minute, Star? Because
Sure.
I think that's a key question. I think that if you were to kind of separate the individual human capital, the kind of quality of scientists in the national programs, and compare it to 20 years ago, it's pretty clear that the individual scientists are better trained. But if you would compare the institutional capacity to deliver genetic gain within NARS compared to 20 years ago, I think you would have a much more mixed picture. It's not at all clear that the NARS today are stronger in their capacity to deliver than they were 20 years ago. The picture that emerges is they're not releasing more varieties. They're not doing more crosses than they were in the past. If you, again, separate out the quality of the scientists that are in staff, in the CG and in the NARS, better trained, the individual scientists within the NARS have great incentives to build their human capital and then move on to another, build their career, not necessarily within the NARS, move to the CG or some other institution. And so as institutions, the national programs are very unstable. Funding is not secure. Resources are not necessarily growing. We have very poor measures of those. And the CG has been incredibly unstable over the last decade. So I think it's very important to look at developing some measures of institutional capacity and stability, keeping in mind that capacity of individuals is an important component, but it is not always going to solve the institutional stability problem and lead to more effective delivery of genetic gain.
Yeah, thanks, Greg, for that addition. The huge challenge is how do they really, the problem so much greater than the resources they have. It's very difficult to do what they are asked to do with 3 full-time people, and maybe they do not have the expertise, and they need to have the resources, infrastructure, etc. And then people work in NARS, this national level, I think they are only at maybe 1/3 or 1/4 of the salary as the same, like say, classmate who went to work at the CGIAR scientist. So Jiping, maybe the last question, and I can stay for other people have question. Maybe Sage and Greg can stay as well. Last question. Yeah.
Yeah. So, okay. So you done the survey. I don't know what's your plan. I mean, you can give the feedback to different institutions. What's the plan?
Yeah.
What the next step? I mean, you have the survey, you have the data. I guess your goal is of the surveys are to give some feedback to the different institute, right, or reader or.
Right. So my part of the survey, I can speak to, then Sage and Greg can answer to that part. So my part of the survey, after we finished, we shared the results with the key leadership, right? That's the justification. These the functionalities we need to put together. These the priority, and then to build this one centralized analytic services, prioritize which one to go, and that's where we left with. And then this May, we even brought all the analytic team together in Egypt. And that's what we did at the planning, phase by phase, what functionality we focus on, the components in the matrices serving this entire pipeline. So this is one thing, internally deliver this minimum viable implementation, connect with databases. So with the, like say, Brabi or data lake, connecting between the different databases with the functionalities they are asking. This is one thing. Another thing is the first part of the Go 1 part survey I didn't present, but this is also asking the breeding program, are you willing to contribute and what percentage of the budget to support this centralized analytic services and support? And each of the service support, we have them as service have an order number, just like you order genotyping, you have a price. This may have a price, or this may be just as a service, as a support. This come from the breeding budget, but you don't have to pay for. So this where we were trying to move on to the new operational model. That's the survey background. And we didn't get there because the funding, as some of you are already aware, and hopefully new phases in 24, different story that can continue building this. So how about for Sage, Greg, would you like to add? Did you hear Jiping's question? After the survey, where do you take to the next stage? Do you give back to the participants of the survey?
Why don't you go ahead, Sage?
Sure. So our plan at the moment, again, we're conducting the kind of same measurements at the end of this project. So we took the initial baseline survey in 2020. The first thing that we did is we shared a lot of these results with the ILSI objective areas, and so that we could provide kind of this baseline information. So they knew within their kind of relevant metrics where the breeding programs were at that they were working with. And so this upcoming year, when we take these measurements again, the plan is to have different kind of reports available, and we'll meet individually with the breeding program leads and whoever can join the call and give some survey feedback. Another thing, like I mentioned, we've been visiting a lot of the breeding programs. So far we visited 4 of them, with a plan to visit in this next year 3 more. So that's an opportunity for us to give some feedback and talk about these. And then hopefully, this is still up in the air, but if there's, another cycle of this project, this could be involved in the planning process, moving forward, how to take these kind of gaps in institutional capacity and really plan for, the next 5 years.
Yeah.
And we've also shared our results with the national programs to allow them to place their own program in context of peer programs. So they can kind of see who's doing what, who's using what capacities exist within other NARS.
Yeah. And also just by working with Sage and Greg and Mathew the last several months, we are still collecting. Basically, we have an ongoing survey, and in January when I go to PAG, we'll continue to collect data from the participants, and these insights is going to help us to plan the next phase. It's about matching the demand with the supply and identify the opportunities and the challenges where, and also if you do customize the support at an individual center or the stages level. So this give us this as a foundation to be more tailored for next phase. Okay. All right. Thank you so much. I really appreciate it by working this semester and then answer the survey from the graduate school or from the course for evaluation, and have a great final. Yeah, thanks. Thank you, everyone online. Have a good day.
Thank you, everyone.
Yeah.
Pleasure.
Thank you so much, Sage and Greg. Yeah.
This has been a production of Cornell University, on the web at cornell.edu.